Okay, so we're going to go a little bit more in depth with our carbohydrates particularly and just have a little bit of extra information about our lipids. So last time, previously in IB Bio, we talked about different kinds of sugars, some of which you learned how to draw in depth. So if I show you this molecule with this sort of up, down, down conformation in a six-member ring, you can probably guess that this is glucose. And another important sugar that we learned how to draw last time, which was that five-membered ring, which remember the sort of the pizza guy conformation. This was ribose. So each of these are considered single sugar units. And in science, sugars have a fancy name of saccharide. And so since these are single sugar units, they are called monosaccharides. So that's just a single monomer of a saccharide. So glucose and ribose are both examples of a monosaccharide. So there are a few other monosaccharides that we have to be familiar with. And we'll just go through those here. So one of them is galactose. So galactose is found in high concentrations in things like dairy and sugar beets, and as you can guess from the picture, some honey. So galactose is very similar structurally to glucose, right? So if glucose, we have this up, down, down conformation, galactose is almost exactly the same, except that that first hydroxyl group is faced up instead of down. So that's the only difference between glucose and galactose. So both of them are six-membered carbon rings, and both of them start with this CH2OH group in the up position, but glucose, the hydroxyl group is pointed down, and galactose, it is pointed up. So that's how you distinguish between them. So the other monosaccharide is going to be fructose. So fructose, as the name suggests, is going to be commonly found in fruit. It's what makes apples and strawberries sweet. So structurally, fructose is very similar to ribose. It's another five-membered ring. But the differences is that one of the so-called lake positions is going to be in the up position instead of down, and that we have this addition of a CH2OH group instead of a hydroxyl group here. So that's what makes fructose different from ribose. So again, just to review, your monosaccharides are glucose with this sort of up, down, down conformation, and galactose is different from glucose in that we have, instead of this hydroxyl pointed down, it's pointed up. Ribose is going to be that five-membered ring. This is sort of the dude carrying the pizza. Here's his head, here's his 2 feet, and he's got his other hand in the air. And then fructose looks a lot like ribose, except that this hydroxyl group is pointed up and it's got this extra CH2OH group. So that's how you distinguish between ribose and fructose. So remember that one of the important features of carbon molecules is that they can be linked together, right? Carbon likes to form 4 bonds, and you can link those carbons into long chains. So by extension, sugars can be joined to form long chains too. So let's start small and talk about linking 2 monosaccharides together. So mono means 1, di means 2, so disaccharides are going to be 2 of these sugar molecules joined together. So a disaccharide, again, is just 2 monosaccharides that are going to be linked together. And like all anabolic reactions, these occur via condensation reaction. When they join together, they're going to release water as they're building that molecule. And the linkages between these disaccharides come in 2 flavors. They're going to be either glycosidic or galactosidic, and that just depends on the molecules involved. If it's glucose and glucose, it's glycosidic. If you add a galactose in there, it's the galactosidic, so it's pretty straightforward. And the linkages are also named by the carbons that are involved. So I believe this is going to be carbon 1 and that one's 4. So the first disaccharide that we're going to talk about is maltose. So maltose is glucose and glucose linked together. So here is one glucose molecule that is linked to a second glucose molecule by a 1,4 glycosidic bond. They sort of go down in between them here like this. So that's maltose. So as long as you see 2 glucose molecules linked together, you know you're looking at maltose. The other one is sucrose. So this is what makes your cakes and donuts delicious. And this is a glucose molecule linked by an alpha glycosidic bond to a fructose molecule. And so this is going to probably be the easiest one to distinguish because it's going to be the only one that you see with a 5-membered ring in it. It's got that fructose again with the upper hydroxyl group and that lower CH2 in the bottom there. So lactose is a disaccharide that you're probably familiar with. So like I scream, you scream, we all scream for ice cream, especially if you're lactose intolerant, you're going to have a bad time for this, right? So lactose is glucose, regular old glucose, linked to a galactose. Now, this sugar is commonly found in dairy products, right? And again, it's galactose plus glucose. But this bond between this disaccharides are going to be a little bit different, right? So this is a beta-galactosidase linkage, so they kind of stack up a little bit. So a lot of people do not have lactase that's going to break this bond, so they can't break down that sugar. So that's what results in lactose intolerant. So even if the human that ingested that, ice cream can't break down this lactose, doesn't mean the bacteria in them can't. And so when that happens, it creates a lot of gas in the person, makes them really uncomfortable, and we know that as lactose intolerant. Okay, so just to review, your disaccharides are maltose, which is glucose and glucose linked together in this glycosidic bond. Sucrose, which is going to stick out because it's the only one that you see that has this 5-membered fructose ring in it. And then there's lactose. And the only difference between maltose and lactose is going to be this galactose. So how you tell between them easily is probably by this weird bond, this galactosidic bond right here, which is going to sometimes be shown staggered or stacked. And then the other place you want to check is in these hydroxyl groups. So if it's pointed up, you're looking at a lactose, versus if it's down, you're probably looking at maltose. So those are your disaccharides. So if you can link two monosaccharides together, then you can also link many. There's no reason energetically that you can't string a bunch of these together. And in fact, that happens quite often. So polymers of monosaccharides are going to be called polysaccharides. So poly just means many, so many sugars. So polysaccharides come in a lot of different forms, and they can have a lot of different jobs. So one of them can be structural, right? So in the case of cellulose in plants, what gives them that rigidity is the cellulose. They can also be used as fast or short-term energy storage in both plants and animals. So the three kinds of polysaccharides that you're going to, or I guess four, that you're going to have to distinguish between are going to be cellulose, glycogen, which is in animals, and starch, which comes in two forms, amylose and amylopectin. So all of these polysaccharides that we're going to be looking at, all of them are just polymers of glucose. It's only the linkages that are different between them, and that's going to give them really different structures. So let's start with cellulose, and you can probably guess from the picture that this is going to be a component, an important component of plant cell walls. So cellulose is what gives plants their cell walls that rigidity. So they have, more structured cell shapes, and they're more rigid than what you find in animals, and that comes from cellulose. So what cellulose actually is it's going to be, these linked stacks of glucose molecules, which gives it, makes it very sturdy, because you've basically got this brick wall of cellulose. So humans cannot break these bonds in cellulose, so that's why a cow can eat grass for energy, but you can't, because we can't free these glucose molecules from each other. So only certain animals, only the herbivores are going to have the enzymes to do that. So in humans and other animals, we have a molecule called glycogen. And so glycogen is for fast short-term energy storage. And again, this is just glucose molecules that are being linked together. But what you can see is that glycogen is structurally very different than than cellulose, right? So cellulose had these, stacks, the sort of brick wall structure, but glycogen is highly branched structure that you see here. And again, all of them are glucose. So glycogen, cellulose, and starches are all polysaccharides of glucose. So the other polysaccharide that you're going to have to know about is starch. So starch is going to be the energy storage that's found in plants. It's what makes potatoes so delicious. Why you love them is because this is basically a sugar. So there are two forms of starch that you have to know about. One of them is amylose, which is going to have this sort of spirally structure. So if you see that, that is amylose. You can stop right there. The other one, which might be a little bit more tricky, is going to be amylopectin. So this one has a more branched structure, And so the problem is going to become when you're trying to distinguish between glycogen and amylopectin. And so they're going to give you some sort of hint. If they say it's a starch that's branched like that, it's going to be amylopectin. If they don't say anything about starch and it's, branched like this, then you're looking at a glycogen. All right, so just to review, your polysaccharides are cellulose, which has this highly, brick wall structure. It's what gives cell walls rigidity in plants. It's a structural function. It's just there to like build a wall. And remember, humans cannot digest this stuff, only the herbivores, because we can't break these linkages. So the other polysaccharides that you have to know are the two starches, which are energy storage in plants. And remember, that comes in two forms. Amylose, which is just like this thread sort of spirally structure. You might see it as like a series of loops, a real spiral structure. Or you might see amylopectin, which is going to have these branches. And then lastly, we have glycogen, which is the energy storage in animals. So this is fast energy, right? And it's going to have that highly branched structure. So this one is pretty easy to distinguish from cellulose. Okay, so for the lipids, I really only wanted to add one more kind of lipid, and that is the triglycerides. So triglycerides are going to be the main energy storage in animals. So glycogen, yeah, we do use it, but it's only for temporary quick use, kind of on a daily basis. Triglycerides are more for long-term storage. That's what's going to be stored in your fat cells, around your belly. This is how energy is stored for, on the order of months or weeks. So triglyceride, as the name implies, involves three somethings. And the three somethings is three fatty acid chains. So remember, your fatty acid has this carboxyl group on one end and a methyl group on the other end. And so there's just three of them that are stacked together. And they're going to be attached to this glycerol molecule. So this is what a glycerol molecule looks like. And if we look at what's in between them, you can already guess that water is going to be released. So again, this is an anabolic condensation reaction. So when these linkages are made, those waters get kicked off, and now we have this triglyceride or neutral fat. So triglycerides, again, are going to be that slow, long-term energy source. This is what gets stored in, your fat cells, in your adipocytes. And triglycerides, pound for pound, store way more energy than carbohydrates. So about 160 times more energy than a carbohydrate. So you distance runners out there, once you burn through your glycogen, when they're doing those marathons and they hit like mile 17, they switch from burning carbohydrates to burning triglycerides, and that's kind of why people feel so terrible. So yes, there's a lot of energy storage in there, but it's not as readily available as quickly as what you have in the carbohydrates. So again, slower, longer term energy source, but definitely holds more pound for pound. So while on the subject of fats, right, so we have this not so great way of calculating, how much fat is too much fat on a person. And so we're going to talk about calculating your BMI with something called a nomogram, right? So BMI, or the body mass index, is basically a ratio of your height to your relative fluffiness, so how much you weigh in pounds or kilograms, and that gives you a number called the body mass index. So anything under 25 is considered to be healthy, and anything over 25, maybe time to switch to some veggies, some carrots, and time to hit the gym. So this is an imperfect system. It sort of can label people who have a lot of muscle on them as being unhealthy, even though they're not, but it's, what people have used for a long time. It's kind of rudimentary. So this picture right here would be called a nomogram. So this chart is what people can use to find a BMI. So how you use it is we have weight on the left here and height on the right here. And in the center is, where they're going to say, okay, this is where the body mass index is. So how you use this is you find a weight on the left. So say, and let's just do this in pounds because we're in America. So let's say we have a 140-pound person who is, say, 5 feet tall. So that's going to be somewhere in here. So you would draw a line between 140, where is that? So 140 and say 60, if it's up here, right, because we're doing in inches, then where this crosses is going to be their BMI. So let's say we have a patient who weighs 130 kilograms and they're going to be 65 inches tall. So wherever this line crosses and it looks like it's about 41, 42, that would be the BMI for the person. So that's how you, that's how you calculate BMI. All right, that is it for today.